take it away from here. Thank you very much for being here.
All right. May I see my slides? Right. So I'm going to try to review the state of the vaccine world. First, very briefly, from the point of view of history, and then secondly, from the point of view of the future, or at least the current, present, and the near future. Next slide. So these are conflicts. Next slide. So, you know, we are in the midst of a pandemic, as everybody knows, but I think it is well to realize that this is not the first pandemic in the history of the world, and that indeed we can expect there will be future pandemics, so-called disease X. Because if you look back, you can see, and these are only the major epidemics of the past, you can see that humanity is subject to periodic epidemics and pandemics, and this will certainly continue. Next slide. What has made the difference is vaccination. As we've said before, the impact of vaccination on the health of the world's people is hard to exaggerate. With the exception of safe water, no other modality has had such a major effect on mortality reduction and population growth. Parenthetically, when I was a child, I had 3 life-threatening diseases from which I fortunately survived. But that was not unusual, and that was only 90 years ago. So we should be aware of this history. Next slide. And again, this is a diagram, if you will, a map of the United States in 2006 with respect to rotavirus outbreaks in infants. Which could dehydrate and kill infants. And fortunately, my lab and one other lab developed a rotavirus vaccine. And the next slide shows the result of that 10 years later. And so this is just another example of the value of vaccines. Next slide. So, of course, we all know that vaccination began with Edward Jenner, with essentially the use of a animal virus to protect against a human disease. Next slide. But nothing much happened for many years until Louis Pasteur began to systematically attenuate viruses and also bacteria, for that matter, and this, of course, is a figure of Joseph Meister being vaccinated against rabies. So, in a sense, definite sense, that was the first vaccine to be developed in the laboratory. The next slide shows an important other vaccine, and that was Bacille Calmette-Guerin. And the reason I stress that is because that was the first really attenuated vaccine, that is taking Mycobacterium bovis and passaging it to attenuate its virulence. Next slide. But it was cell culture developed in Boston that really opened the way to attenuating other agents. This is a picture of John Enders and Sam Katz, who unfortunately just died some days ago. where they developed an attenuated measles virus on the basis using cell culture in order to attenuate. Next slide. So if we look at attenuated vaccines in the past, we can see beginning with vaccinia, physical attenuation, and then passage in animals, eggs, or in vitro, giving us a number of vaccines, including MMR and rotavirus and live influenza vaccine. Next slide. We have now ways of developing new attenuated vaccines, temperature-sensitive mutations and reassortment is a valuable way of attenuating a virus. And I would stress codon de-optimization also. This is, for example, we now have, at least experimentally, a codon de-optimized COVID vaccine, which at least in early studies looks interesting. And we can use live or non-replicating vectors to present genes from pathogens. Next slide. Inactivated vaccines actually, if I may so, began in the US when Daniel Salmon, who of course gave the name to Salmonella, and the next slide, Theobald Smith, first working together, first inactivated bacteria. And of course, a number of important bacterial vaccines, typhoid, cholera, followed that path to prevent serious diseases. Next slide. And of course, some bacteria produce toxins, and Gaston Ramon developed ways of inactivating the toxins using formalin, giving us diphtheria and tetanus vaccines, which certainly have saved millions of lives. And the next slide shows Oswald Avery at the Rockefeller Institute, who showed that you could use bacterial polysaccharides to immunize, but if you conjugated them with proteins, they were even more immunogenic. And of course, later on, it was shown that that would be important for vaccinating children. Next slide. But this brings us to the current era. Herb Boyer was one of the inventors of genetic engineering, using genetic engineering to take antigens and to convert them into DNA or RNA, and to change their coding and to make essentially new vaccines, new inactivated vaccines. Next slide. So the history, as I said, of inactivated vaccines begins with kill whole organisms, development of toxoids, extracts, polysaccharides, conjugating those polysaccharides, and of course purifying proteins, and now producing those proteins genetically. And that gives us important vaccines. And the next slide shows that these strategies for inactivated vaccines now include, as I said, the capsule of polysaccharides, but also reverse vaccinology, that is starting with the genome and identifying proteins produced by the genome, structural analysis, and I'll come back to that point. And of course, nucleic acid vaccines, both DNA, messenger RNA, and self-amplifying RNA. And the next slide. So if we look back then at the history of vaccine development, we can see that the cumulative number of vaccines developed is rising. And this, of course, gives us hope for the future. So the next slide is simply to point out that we are now looking at many different ways of using the immune system to immunize, not just neutralization and cytotoxicity, but using other properties of antibodies and cells, binding antibodies that prevent attachment as well as neutralization, Th17 cells that attract neutrophils and help prevent carriage of organisms like pneumococci. Antibody-dependent cellular cytotoxic antibody may or may not, depending on whom you believe, be useful for HIV. But in any case, natural killer cell activity is clearly important, an important immune factor in many diseases, both viral and bacterial. And of course, stimulating T cells, both CD4, as in the case of pertussis, and CD8 in the case of probably of COVID. These are important. And the next slide points out that our knowledge about adjuvants has increased recently. We now have quite a library of adjuvants, whereas before aluminum adjuvants were essentially the only ones we were really using. But now we have a whole library of adjuvants, TLR stimulators, and as this list shows, a number of different powerful adjuvants. And I should have put on this slide CpG, a TLR9 agonist, which is being used now in several different vaccines, and which is really increasing immunogenicity markedly. So the next slide actually is about TLR adjuvants and a whole variety that you can see here, stimulating different pathogen recognition receptors, but each one finding some use, at least experimentally, and in some cases licensed vaccines. So now I want to focus on a few current vaccine developments as examples of what is going on, so to speak. Next slide. Now, with respect to influenza vaccines, as you know, we have both live and inactivated influenza vaccines, but we have problems. The inactivated vaccine is less effective in children and the elderly than in healthy adults. The live vaccine that is very useful in children is ineffective in the elderly. And both types of vaccine are less effective if the circulating virus changes from the previous year. And if we're honest, vaccine efficacy is only moderate at best with our current vaccines. Next slide. So one important point is that the hemagglutination inhibition titer, which we usually use as a correlate of protection, is really not sufficient. Microneutralization, for one thing, may be a better way of measuring the efficacy of influenza vaccines. But in addition, using the hemagglutinin alone is clearly insufficient. We need to add neuraminidase, for example, and other parts of the influenza virus apparently could increase protection. And with regard to measuring antibodies, we should also be looking at FC effector functions like ADCC, because those antibodies also appear to contribute. And then, our inactivated vaccines are not doing anything with regard to mucosal antibody. And at least for the live vaccine for children, it is clear that mucosal IgA is an important protective factor. And lastly, why are our vaccines, influenza vaccines, so poor in the elderly? And one possible reason that certainly has been advanced by workers is that T-cell function, CD8 T-cell function, is important in the elderly, and our vaccines are not inducing those responses. So a lot can be done and is now in course of being done to improve our influenza vaccines. Next slide. Well, this is just the issue of neuraminidase, which I've just emphasized. The next slide. And now there's a lot of focus on moving away from the head of the influenza hemagglutinin to the stalk, shown in blue on this cartoon. The point being that responses to the stalk appear, one, to be broad, and second, to markedly increase the efficacy of influenza vaccines. Now, this is the practicalities of this are still being worked out, but I do foresee that we will be adding other parts of the influenza virus, not just simply the head, for the in the future. And the next slide shows two approaches to that. One is to use two different heads, but the same stalk, to try to induce antibodies to the stalk. The more obvious approach is to use headless hemagglutinin, but that doesn't seem to work as well as the chimeric hemagglutinin approach. So we may need to have 2 doses of influenza vaccines to give the broadest protection. Next slide. So moving now to another important virus, respiratory syncytial virus, and the value of structural biology as an integral part of vaccinology. Next slide. Respiratory syncytial virus, as you know, is the most important respiratory infection in infancy, but is also important in the elderly, second only to influenza. And the history of vaccine development was marked by an episode where inactivated RSV actually worsened disease. And the reason, it turns out, was that the fusion, the F antigen of the virus, the surface of the virus, which is extremely important in producing antibodies, was altered in prior attempts to develop vaccines, leading to formation of immune complexes and therefore illness in infants who received the RSV vaccine. Also, attempts to develop live attenuated RSVs failed because basically the mutants were insufficiently attenuated, especially for the youngest infants. So a significant problem in developing RSV vaccine. Fortunately, next slide, Fortunately, people have continued to try to develop RSV vaccines, and I'm not going to go into detail in this slide, but in addition to attenuating RSV or trying to attenuate it using vectors, using monoclonal antibodies, which does work, in fact. We now have long-acting monoclonal antibodies. Nanoparticle approaches and subunit approaches have all been tried, but the signal discovery, next slide, was that the F protein was changed during the preparation of RSV vaccines based on the F protein. So in the middle is the cartoon of the structure of the prefusion F, and it was almost always converted to postfusion F in the production of or attempted production of RSV vaccines. But if you stabilize the prefusion molecule, there are at least 4 epitopes on the F, which is productive of protective antibody. So this type of prefusion F is now in active development and is looking very good. Next slide shows that neutralizing antibodies are produced in children and adults if you use the prefusion molecule as your antigen, and this shows serum antibody. But what is not clear is this the protective antibody, or next slide, is it that this type of vaccine can induce mucosal antibody on the surface of the respiratory mucosa, as shown here, both IgG and IgA? I think it is likely that both types of responses are important in the efficacy of these new vaccines, which has been reported to be better than 80%. So we can certainly anticipate a licensed RSV vaccine in the near future, at least for children and possibly for adults as well. Next slide. So a virus that I've paid a lot of attention to myself is Cytomegalovirus. Next slide. Cytomegalovirus is, in the absence of rubella, which is now being controlled by vaccination, is the most important cause of congenital abnormalities if it infects a pregnant woman during the first and early second trimester of pregnancy. Attempts to develop a vaccine actually started, if I may say so, with myself something like 60 years ago. So it has not been easy. But the molecules that seem to be important for CMV are shown on this slide, starting at the bottom. Oops, go back, yeah. Well, starting in the middle, the glycoprotein B on the surface of the virus induces neutralizing antibodies. Also, at the bottom, there is a pentamer, that is to say, 5 different proteins which form on the surface of the virus a pentamer, and those proteins are there: GHGL, UL128, 120. 130 and 131. Anyway, so these are important in inducing neutralizing antibodies. At the top are the 2 molecules that are important in producing T cell responses, the tegument protein PP65 and PP150. Next slide. The interesting thing, or one of the interesting things about CMV, is that seropositive women are not totally immune to having an infant infected in utero with resultant abnormalities. The issue is this, in my view, and that is that CMV has the property of becoming cell-associated very rapidly. As soon as infection takes place in the nasopharynx, the virus is able to infect cells and circulate as infected cells rather than cell-free virus. Now that's true, let's say, in the average child. Most children eventually develop CMV infection. But if a woman is seronegative, if she grows to the age of having children and is still seronegative and becomes infected, she has, at least temporarily, cell-free virus, which obtain entrance into cells and become, the woman becomes chronically infected. But it's at that point, when she does have cell-free virus, that the fetus appears to be infectable and may develop severe abnormalities. But what about seropositive women? Well, they are relatively resistant to superinfection, not entirely, but if they are superinfected, they have cellular immunity, which, that is to say, immunity which is able to destroy infected cells for the most part. But indeed, a percentage of seropositive women can also bear infected children. The protection, that is relative protection between of seropositivity versus seronegativity, is between 75 and 90%. So natural immunity does give them some protection, but is not complete. Nevertheless, vaccinating seronegative women, converting them to seropositive, would be valuable, to say the least. Next slide. And so a lot of work has been done trying to identify protective factors, and these are listed here. High antibodies to the pentamer, as well as to the GB, developing a lower viral load after infection, in contrast to a high viral load, and cellular responses, both CD4 and CD8. So the immunity to cytomegalovirus is extremely complicated, but vaccines now are in late development, and the idea is to convert a seronegative woman to a seropositive woman so that she has at least, let's say, 80% protection against congenital infection. Next slide. I just wanted to say a word about dengue, because it's an interesting example of correlates of protection. Next slide. That is, we have a vaccine, but it's not a great vaccine, because the protective rate is low and only seems to operate in individuals above the age of 9. And what is the problem? The problem is that you have 4 different dengue viruses, and the vaccines that are being developed are supposedly containing antigens against all 4 viruses. But the problem is that, and this applies to all of the current, let's say, vaccines that are being developed, is that only 1 or 2 of the 4 components gives homotypic antibodies. And apparently one needs homotypic neutralizing antibodies for efficacy against each of the 4 serotypes. So the effort now is to try to develop a quadrivalent vaccine that does indeed give homotypic responses to all of the 4.
Stanley, could you please unmute? It seems like you've muted your computer.
Sorry, I don't know how that happened.
Okay, we can hear you again.
Okay. Next slide. Just to say a word about SARS-2, the cause of COVID-19. Of course, all of us have been very much involved in studying this virus in the last few years for obvious reasons. This, of course, is a cartoon of the virus showing the spike protein. But I also want to emphasize, because this is part of current vaccine development, that the spike protein is not the only protein in this virus. There's the membrane glycoprotein, the envelope glycoprotein, and of course the nucleocapsid protein. And these are all important in producing immune responses. And I think in particular, the nucleocapsid protein, which is important in inducing cellular immunity, has not been given sufficient attention. But anyway, next slide. What is new about vaccination for COVID-19? Well, I think it's important that the public wants vaccination. This is an important point for the future of vaccines. Secondly, that multiple strategies have been used for vaccine development. We've learned an awful lot about vaccine development in the last 2 years. Third, that governments are supporting financially the development of vaccines. Fourth, of course, the importance of mutations, which should have been obvious. And lastly, that there are multiple manufacturers, including in lower middle income countries. This is a revolution for vaccine production, a good revolution. In the future, manufacturers throughout the world, including in lower income countries, will be able to produce vaccines. Next slide. I want to just stress a couple of points with regard to this virus. One is that in studying correlates of protection, of course, you hear a lot of stuff about this and that, but in fact, neutralizing antibodies are the major correlate of protection. As shown in this slide, the x-axis showing the neutralization titer, the y-axis showing protective efficacy, and it's the vaccines that produce high levels of neutralizing antibody that are most protective. That's not to say that other immune responses are important, for example, in persistence of protection, or with respect to cellular immunity in protecting you if you do get infected. That is to say, T-cell responses modulating your infection. But the main protective factor is antibody. And the next 2 slides point out, next slide, what you certainly know, and that is the other positive thing about this experience is the large number of strategies that have been used to develop vaccines against SARS-2. And this bodes extremely well for the future of vaccinology. All the vectors that have been tried and which have given fairly good results. Not everyone, but many of them. The next slide. even the protein subunit nanoparticle vaccines. These are working, and of course, needless to say, DNA and RNA vaccines have come to fruition in this in these terrible last few years. So a positive result of a terrible experience. Next slide points out that reverse vaccinology is a way of developing bacterial vaccines by identifying the important genetic component that codes for a protein that is protective. And this method has been used particularly for meningococcal group B vaccine, but is certainly going to give us other bacterial vaccines. Next slide. Well, this just gives examples of vaccines that have been developed using reverse vaccinology, sorry. And we will see some of these being licensed without doubt. Next slide. And new attenuation techniques will enable us also to develop better vaccines in the future. Codon de-optimization and controlling replication fidelity will both be useful. Now, in the interest of time,
I think you're muted again, Stan.
I don't know. I'm not doing it. I don't know what's happening. But anyway, so new attenuation techniques will help us to develop more attenuated vaccines. Next slide. And tuberculosis is an important target for vaccine development. And just to say that using vectors, virus vectors, with insertion of genes from Mycobacterium tuberculosis is now being explored. Next slide. And of course, you all know that malaria vaccines have been developed, are being used, are giving at least a moderate protection, and if used repeatedly will certainly prevent serious and fatal malaria. And let's move past the next couple of slides, and just to conclude, I realize that much of the meeting that you were attending, previous slide, much of the meeting that, I'm sorry, go ahead, next slide, and next slide. Oops. Yeah. So, of course, I know that much of the meeting that you were attending concerns so-called non-specific protection. And as an aged vaccinologist, I just want to say that I have no doubt that non-specific effects, or whatever you wish to call them, is a real thing. And it is a property which certainly relates in particular to live vaccines that stimulate responses that go beyond antibodies and specific cellular immunity against the agents themselves. This slide speaks to the reduction in mortality when BCG or measles vaccine is given. And of course, as you all know, much of these results have come from from where you were meeting now in Denmark, the work of Peter Abbey in particular, and certainly one should be proud of the fact that this idea of the importance of immune responses that go beyond the specific immune response, this idea is of extreme importance and is being applied now to many situations. And I just, the next slide is the result of a study, an interesting study by Faustman et al., in which BCG, in contrast to placebo, had an effect on COVID-19, on the seriousness of COVID-19 infection. So this is a part of vaccinology that needs to be explored a lot more, to be applied, particularly in low-income countries, to reduce mortality. So I want to conclude, next slide, by saying that there are still unsolved problems in vaccinology. For example, immune memory, which is a very big problem for the messenger RNA vaccines, but also in complex pathogens, particularly bacteria, the multiplicity of virulence antigens is something that needs a lot of work. And structural biology is enabling us to use epitopes that in vaccines that were not possible before. Systems biology is helping us find correlates of protection. And the last slide, we still are not good at inducing responses in the very young and the very old, and we need to find better ways of doing that. And mucosal immunization with non-replicating antigens is certainly important if we're going to stop viruses and bacteria from spreading, even if they don't cause disease. Adjuvantation, as I mentioned, is certainly important. And the generation of T-cell immunity as well as B-cell immunity is something we need to improve. But I am optimistic about the future because of what we've accomplished in the past. Thank you. Hello?
Not on. Yes. Sorry. Thank you very much for being with us today. And no, you couldn't be here in person, I think, because you were receiving a well-deserved prize in the US, but we were so share your knowledge on non-specific or specific and non-specific effects of vaccines with us today. I believe in the interest of time, we will move on to the next speaker.
Okay, well, thank you very much for listening.
Yeah. Thank you very much again. Yeah. So